Hello everyone, and welcome back to part 2 of MCP 102 review. So we will continue off from part 1 by talking about rotational angles, about the various bonds in the polypeptide backbone. To recap, the omega angle is between the nitrogen and the carbonyl-bearing carbon, and is restricted to either 0 or 180 degrees, while the phi angle is between the nitrogen and the alpha carbon, and the psi angle, which is between the alpha carbon and the carbonyl-bearing carbon, are both able to freely rotate at any angle. However, due to steric interactions, certain angles are more stable and are primarily seen. So, the Ramachandran plot is a graph that shows the various combinations of the phi and psi angles and identifies the combinations that form the typical secondary protein structure features, such as alpha helices and beta sheets. Now, a secondary protein structure is formed by interactions between the polypeptide backbone, which is primarily hydrogen bonding between the nitrogen atoms and the carbonyls. So the side chain interactions do not come into effect at this level of protein structure. So on the Ramachandran plot here, the red regions indicate the most commonly found angles for their specified structures, whereas the yellow regions indicate the full range of allowed values. Now, alpha helices are predominantly right-handed, but it is possible for left-handed helices to occur. So a right-handed helix in proteins occurs naturally because a right-handed helix is formed from L amino acids, which are the primary amino acids found in animals. Now, one turn of the helix on average includes 3.6 amino acids, and each turn is separated from an adjacent turn by 5.4 angstroms. Now, proline and glycine are the two amino acids that tend to not be found in alpha helix patterns because their unique structures cause them to break helix patterns. Beta sheets on the second type of secondary structure and are produced when strands of the polypeptides run parallel or anti-parallel next to each other. In a parallel beta sheet formation, the strands are all running in the same direction, while in the anti-parallel formation, the strands alternate running in one direction and the other direction, similar to how the strands in DNA run in opposite directions. The anti-parallel beta sheet is a more stable configuration since it allows nitrogen atoms and carbonyls on adjacent sheets to align more closely and form straighter and thus shorter hydrogen bonds, which make them stronger and more stable. The key takeaway regarding secondary structures is that they are determined by the interactions between the backbones. So this means that side chain interactions do not affect secondary structure. However, at the tertiary structure of protein folding, we now consider the interaction between the side chains. So the primary difference between a secondary and tertiary interaction is whether or not it is existing between a backbone or a side group. Now, some examples of tertiary interactions are the hydrophobic effect, which is when hydrophobic side groups are unable to form stabilizing hydrogen bonds with water around them. So the water molecules and other hydrophilic groups form favorable interactions with each other, and they exclude the hydrophobic side groups, causing those hydrophobic side groups to appear to clump up together in order to reduce their surface area for interactions with hydrophilic or water groups. Second tertiary interaction is the ionic bonds, also known as a salt bridge. So this is just a positively charged side group electrically attracting a negatively charged side group. Then this is why it's another example of why it's important to remember your amino acids and their structures and properties. Another type of tertiary interaction is the disulfide bond, which is when two nearby cysteine residues are oxidized, which converts their two thiol groups into a new functional group called a cysteine. Now, this process is an oxidation, and this results in a covalent bond forming that is a strong connection. Finally, we have the intermolecular forces like hydrogen bonds and London dispersion forces that are also able to affect tertiary structure. The final level of protein structure is the quaternary structure, which is determined by how the various subunits of a protein arrange to form the overall structure. Now, by definition, only a protein with multiple subunits can have a quaternary structure. So if a protein is made up of only one polypeptide, it lacks quaternary structure. So at this level, there are many different kinds of symmetry that can be observed due to the arrangement of this subunits. If the protein has two-fold symmetry, meaning that there are two groups, then it's a dimer. If there's three groups, it's threefold symmetry, it is a trimer. And fourfold symmetry, a tetramer, and so on and so forth. Now, a homodimer is a dimer, so it has twofold symmetry, but the subunits in symmetry are the same. Now, if the two subunits are in symmetry are different, then it's a heterodimer. Now, one thing to clarify is the distinction between a protomer and a monomer. So the monomer is the smallest repeated subunit, whereas the protomer is the smallest repeating unit that makes up the protein. So this means that a protomer can be made up of more than one monomer, but a monomer is always only one subunit. So in this case, let's take a look at tubulin, which is a protein that is commonly found in microtubules. So tubulin is the protein which has heterodimers as its protomer, and each protomer is consisting of two monomers, one which is alpha tubulin, other one which is beta tubulin. Now, both alpha and beta tubulin would be considered monomers, but only their dimer would be considered the protomer. And with that, we will conclude this section.